The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 46 of the eLife Podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, do statins make your diabetes worse? Was it lack of oxygen or something else that held back the evolution of life on Earth? And scientists discover the brain pathway behind our ability to lip read. But first, say you want to read a particular paper. In fact, you want to read one of your own papers that you published a few years ago. So you go to the journal website, at which point you're presented with a bill to read what's ostensibly your own work. Sound familiar? Well, unless all your papers are with eLife or a similar open access journal, it probably is. But the frustration this has caused has stimulated the creation of what people are dubbing the Robin Hood of online access. They're called Sci-Hub, and Penn State's Daniel Himmelstein has been studying their impact.
They describe themselves as a pirate database for scientific literature. Maybe you've encountered a scientific paper online and seen the dreaded paywall, where it says, you can't read this paper unless you pay $35. So essentially, what Sci-Hub is like a Pirate Bay for scientific articles.
How did it get going in the first place, Sci-Hub?
So in 2011, someone named Alexandra Elbakian created the resource, and she was a Kazakh graduate student, and she was frustrated with her ability to access a lot of the literature she needed for research. So she set out to create a service that would act as if it were a university and download papers that they had access to provide them to people outside of universities.
And what's the scale of what's in the Sci-Hub archive? How many indexed papers are there in there now?
Yeah, so what our study did was really look at the scale for the first time compared to all of the scholarly literature. And we found that there were 55 million articles in toll access journals, so journals that are paywalled and you probably wouldn't be able to read at home. And 46 million of those were in Sci-Hub's database. So that's 85% of all toll access scholarly articles.
Incredible that was achieved in such a short time as well, isn't it? How did they get hold of all of these papers that were ostensibly behind paywalls, and yet they haven't had to spend millions and millions of pounds to get them?
No, so that is a closely guarded secret, but the general methods are known that they will get the credentials to act as if they're a university. So for example, I'm at Penn and I can see the articles here. So they have to essentially proxy or pretend that they're on the Penn network to get access to some articles. And more recently, the creators referred to something called direct access, where it appears that she's hacked directly into the publisher system. Because we've seen instances where 100% of the articles from a journal are in the database before people could even have requested them.
Is this not just theft by another name? I mean, they're dressing this up as, right, this is to empower the average person who wants access to scholarly literature, but actually they've just stolen it.
Depending on your view, it definitely could be theft. Another viewpoint is that the public has funded this research and it benefits society. Almost all the stakeholders would like the research to be public and the papers to be public. So it's sort of just an unfortunate way that the publishing system is that these articles are paywalled in the first place.
And is it getting a lot of uptake?
Yes, it seems to be growing. So we have a few ways of assessing that. We've seen the Google searches for Sci-Hub have really exploded. Every time a publisher sues Sci-Hub, there becomes a lot more interest and news coverage and searches. And in addition, Sci-Hub has released some of the download logs. So in the second half of 2017, over 500,000 articles per day were being downloaded from the service.
So why did you decide to get interested in this and then actually try to assess the scale? What was your motivation?
I said, here's a cool data set, But on a deeper level, I'm really interested in scholarly communication and publishing to become more open. So everyone can reuse and reanalyze papers with text and data mining. And the way for that to occur is for articles to be published under open licenses, like Creative Commons licenses. And what our study showed is that Sci-Hub is really going to push the field in this direction, because it's no longer a realistic business model to have paywalled articles and expect people to pay. So things will have to move towards more open models which benefit society.
How did you actually do the study, though? How did you actually probe what Sci-Hub are doing? Because some of the things they're not going to be keen to impart to the public, are they?
Yeah, so we were able to base our analysis really on the list of articles Sci-Hub released, and we compared that to something called the Crossref database, which is a registry of scholarly articles. So with those two pieces of information, we could say how much of all the literature Sci-Hub covered and how much of each different discipline. So for example, Sci-Hub really had great coverage of chemistry, where it had 93% of all the articles. And the worst discipline was computer science, which was 75%, but still that's quite high. Another very interesting thing we saw is we joined forces with the libraries at Penn to compare the subscription access that Penn had paid for with the access that Sci-Hub provided. So we manually evaluated a small set of articles where we found that Penn had access to about 80% of these toll access articles, whereas Sci-Hub had access to 94%. So Sci-Hub is providing greater access than the University of Pennsylvania, which spends $13 million a year on subscription access to journals.
How have the existing publishers responded to this, though? Because it's not beyond the realms of possibility that they'll track down exactly how their archives are being pillaged, and they'll tighten up so that people can't do this. So is there evidence that they are doing that? And how are they generally responding to this sort of exploitation of what was previously their business model?
They're not happy. So Elsevier has sued Sci-Hub. So has the American Chemical Society. Elsevier is a for-profit publisher, whereas the American Chemical Society is actually a nonprofit organization. What's interesting about both those publishers is they're very highly covered in Sci-Hub. So Elsevier is at 97%. American Chemical Society is at 99%. And while they won their suits, there's not really that much they can do because Sci-Hub's based out of Russia. In fact, their suits, in a way, trigger the Streisand effect, where more people heard of Sci-Hub and said, well, if the publishers are suing them, it probably works.
Daniel Himmelstein. One of the major health crises the world arguably faces is the threat from diabetes. This is intrinsically linked to obesity. But why should weight gain lead to an individual becoming insensitive to their own insulin? David James and Daniel Fazakerley at the University of Sydney suspect that when a person becomes overweight, they end up in a metabolic vicious cycle, whereby high energy intake provokes the production of reactive oxygen species in their mitochondria. This depletes the metabolic factor coenzyme Q, leading to even more free radicals and even poorer insulin sensitivity.
Insulin resistance is referred to as prediabetes, and it's a very important condition now because it's one of the major risk factors, not just for type 2 diabetes, but a whole host of diseases, including many forms of cancer. Quite frighteningly, in Australia, there are probably 3 million people with insulin resistance, but they don't know that they've got it. Our research is focused on trying to understand how people acquire insulin resistance. And of course, we're hoping that that will lead us to develop better therapies that could be used to prevent it.
And Daniel, what tack did you take?
We have access to a number of experimental model systems where we can study insulin resistance. And because we have access to so many, we took the idea that if we could look in lots of different models and find common features across these models, we might be able to find a mechanism that may explain insulin resistance. So what we actually did is we took a range of cell culture models, tissues from animal models and human tissue too. And what we found is that enzymes within a particular biochemical pathway were expressed at a lower level in insulin resistance. And this led us down the path to look for a specific molecule called coenzyme Q, which involved in how the cell produces energy, was decreased in all of the models. And the exciting thing was that when we replenished the levels of that molecule in these model systems, we could restore the cell's response to insulin. So they were no longer insulin resistant.
David, do you think this is chicken or egg? Is the coenzyme Q level low because of the insulin resistance, or does the coenzyme Q level end up low and then cause insulin resistance and it becomes almost like a vicious cycle?
That's the big question, and I'll draw upon some new findings that we have to best answer that. This is a new paper that we're just about to publish with Dr. Mike Murphy from Cambridge University. Mike has developed these chemicals that very rapidly induce production of these things called reactive oxygen species. This is one of the molecules in this energy-producing organelle of the cell called mitochondria that we believe is at the heart of insulin resistance. And our hypothesis in our eLife paper is that what happens first is that there is a lowering of the coenzyme Q levels, and that then somehow leads to an increased production of reactive oxygen species, and that subsequently leads to insulin resistance. So that's sort of our working model. So in this new study with Mike, we can show now that we can get rapid induction of reactive oxygen species, no change in coenzyme Q, but we see an induction of insulin resistance. Now, if we zoom back to the eLife paper, we were able to show that when we genetically modulate the cells by perturbing their production of coenzyme Q, that led to an increase reactive oxygen species production. And again, when we replenish the coenzyme Q, the reactive oxygen species return to normal. So putting all of this together, we're pretty confident that we've mapped a linear pathway whereby first you've got the lowering of coenzyme Q. That leads to an increase in reactive oxygen species, and in turn that leads to insulin resistance.
Daniel, where does the coenzyme Q go? Is it because it's not being expressed? Is the gene not sufficiently active, or is the protein being consumed too quickly?
So in some models, it appears that the biosynthetic machinery to make the molecule coenzyme Q is downregulated. But in other models, that doesn't seem to be the case. And it may be actually that the turnover of the molecule, so how well the cell can hold on to the molecule, is actually changed. So it may not be that in all these different systems, that it's the same process leading to a decreased level of CoQ, and it might be different, but it's the change in CoQ levels that's the common factor.
Now, you said that you can supplement CoQ in cells, and this appears to mitigate the effect. People have been trying to dose themselves with things like CoQ for a long time. There's no evidence that it really makes a dent in diabetes, though, is there?
No, that's right. And in fact, this is a really important point that you've raised. It's probably going to be impossible to replenish the levels of coenzyme Q using oral supplementation. It's the coenzyme Q inside the mitochondria that is specifically lowered. You can't just get more coenzyme Q into the body or even into cells. You've got to get it into mitochondria. And we suspect ultimately we have to develop compounds that stimulate the actual synthesis of coenzyme Q. And that could actually be a very fruitful path to therapy.
Statins are associated with diabetes, aren't they? It's one possible reported side effect. Do they have any role in this process?
This is actually a very significant question, and we're quite careful about how we answer this because the coenzyme Q pathway is downstream of the actual enzyme that is hit by statins. And in fact, we've shown in our eLife paper that if you incubate cells, and I have to preface this by saying it's a fairly high dose of statins, but nevertheless, we have shown that you get a reduction in the levels of coenzyme Q, and you also get insulin resistance.
But owing to the dyslipidemia that goes along with diabetes, most people with diabetes are on statins. So actually, are we making their problem worse in some respects?
Again, let me just preface this by saying this is pure speculation, but I absolutely agree. And it's probably also the case that the link between statins and insulin resistance went undiagnosed for a long time, because it stands to reason that most individuals that are taking statins have dyslipidemia, and they probably also have insulin resistance. And I think it's only very recently that people have recognised that the statins may actually be worsening their insulin resistance, which could be worsening their dyslipidemia.
So Daniel, notwithstanding any of that, what sort of avenues does this open for us to explore now you've done this and shown this association?
Yeah, so we're really excited about our work because we think we've now provided some evidence for a linear pathway between the models of insulin resistance we've used, which include things like diet in mice, a loss of coenzyme Q from a specific site within the cells, it's the mitochondria, the production of chemical reactive species in the mitochondria as a result of loss of CoQ, and the induction of insulin resistance. Now, our current work is working on, okay, so how can we target this pathway we've identified to try and provide some therapeutic benefit? So as David said earlier, one of the things we're really interested in is understanding how we can potentially increase CoQ levels in these models. So that's understanding why they're depleted in the first place. And the other arm of our research is understanding the link between mitochondria and the reactive oxygen species and why insulin doesn't work so well in these cells anymore. And if we can identify those links, I think we'll have a much better chance of developing a therapy.
Tremendous work. Daniel Fazakerley, and before him, David James. You're listening to the eLife podcast with me, Chris Smith. On the way, the role of oxygen in kick-starting multicellular life and a new insight into autism. But before that, you've probably noticed that it's much easier to comprehend what someone's saying when you can watch their lips moving. This suggests that there are connections between the brain's visual system and the auditory pathways that decode speech. So what happens to these connections in people who are born blind? The answer is they're enhanced. And if you track the activity in what would normally be the brain's visual areas in these patients, you can see flurries of nerve cell firing that follow the patterns of speech sounds when a person listens. Olivier Collignon.
So one of the main objectives of this research is to try to understand how brain region develops in case of sensory deprivation. And the model we use is to work with early blind people, and more particularly to try to understand all the regions that are typically dedicated to vision, like the occipital cortex in sighted people, develops in early blind people.
So when a newborn baby first pops out, how well developed is the visual part of the brain in terms of its ability to see?
So that is an excellent question. Actually, what we see is that the visual system is already highly organized at day one. As soon as the baby is born, he is born with an architecture that is dedicated in the occipital cortex, the back of the brain, to process visual input. And this highly imprinted organization led to the assumption that there was like a genetic master plan creating an occipital cortex dedicated to vision. But does that mean that this region stays like this in case of sensory deprivation, for example? This is not the case, and this is what we observe. It's a phenomenon that we call cross-modal plasticity. When the typical region in the occipital cortex do not receive their native input, like for example the visual input, they reorganize themselves to process non-visual input, like auditory or tactile input.
How do you know that they're processing those non-visual inputs?
So if you work with animals, for example, you can implant a microelectrodes in this region and record directly brain activity, actually even single neuron activity. And what you will see is that this neuron, which typically fires to flash of lights, for example, in sighted people, they will start to fire to auditory or tactile input in visually deprived animals. When we do that in humans, what we do is that we use neuroimaging techniques. In our study, we focused on another technique, which we call magnetoencephalography, a technique that records the very faint magnetic field that is produced when neurons process information. And this technique has the very beautiful advantage in combining eye spatial and eye temporal resolution. So we can basically, with this technique, address how the brain processes information in space and time.
So talk us through the design of the experiments you did then. Who did you look at and how did you study them?
So we involved early blind people and a group of sighted controls. They listened to short stories while we recorded their brain activity with magnetoencephalography. We correlated the change in brain activity with what we call the envelope of speech, which is the change of auditory energy across time. And while we correlate these two things, basically it's a proof that these regions participate in the sensory processing of the language input.
So this is when you record the seeing, in inverted commas, parts of the brain, you can show that there is also a sympathetic firing there which follows or is entrained to the rhythmic pattern of the speaking that they're listening to. And that's present in both seeing and blind, or exclusively in the early blind people?
So what we see, it's an excellent question. It's enhanced in the blind. It's not that it's not at all present in the sighted people, but it's more there in the blind.
And did the sighted controls have their eyes closed when you did the study to make sure they weren't being distracted by visual stimuli, which might mask the speech effect?
Right. So both groups actually were blindfolded during the experiment. We just also do that with blind people to put them in the exact same condition as the control group.
And your observation suggests that there's some kind of projection from the regions of the brain which decode specifically speech and the pattern of speech, and those projections relay that activity onto the visual areas. Why do you think they would want to do that?
That is an excellent question. Actually, previous studies, notably from our lab, but also from different groups of researchers, have already shown that the occipital cortex participates in non-visual processing in early blind people. I'll give you a simple example. For example, you have in the occipital cortex of sighted people a region that is called V5. It participates in motion processing, visual motion processing. What we see is that in blind, this region specifically dedicated to auditory or tactile motion processing. Like if this region maintained the function, but switch the input. But in the case of speech or language, this is actually at the start more puzzling. Why language would remap in the occipital cortex, right? What is the link between vision and speech processing? And we believe the link is that when we process speech information, sighted people actually constantly integrate visual and auditory information. Even if we don't realize it, we lip read. Therefore, we believe that there is some kind of intrinsic connection or privileged connection between the visual and auditory cortex to integrate the visual and auditory part of speech processing.
Do you think that what you found here is what underpins phenomena like the McGurk effect, where you play someone one sound and show them someone saying a different sound and they hear a third sound? Could this be why?
So this is a very good point. Actually, the phenomenon of the McGurk effect very beautifully illustrates how speech or language is a multisensory percept. And obviously for this phenomenon to arise in the brain, it means that we need to connect auditory and visual input for this integration. And what we believe is that this is exactly these connections between auditory and visual brain centers that are recycled in blind people to basically expand the response properties of the occipital cortex toward sound processing, and in this case, in particular, speech processing.
Olivier Collignon, he's at the University of Louvain in Belgium. The first life on Earth appeared, we think, about 4 billion years ago, And for at least the next 3 billion years, it hardly changed, remaining as simple single-celled microbes. Then, something happened that triggered these single-celled organisms to begin to team up and turn into metazoal, or multicellular life. But what was it? One theory was the amount of available oxygen. Too little would not meet the needs of these more complex life forms, holding back their appearance. So how can one find out if this was indeed the case? At the University of Southern Denmark, Daniel Mills has come up with an intriguing approach. He's been studying some of the most primitive species of life that were the first to split away from the last common ancestor that we all share. If these early ancestors were oxygen sensitive like we are, he points out, then that's very good evidence that it really was a lack of oxygen that held back the evolution of complex life. So what did he find?
We're primarily trying to figure out the historical relationships that existed between the earliest forms of animal life and then the oxygen content of the environments that these early animals lived in. We think the earliest animals emerged around 800 million years ago or so.
And the environment that they're evolving in, what were the conditions like at that time?
Well, that's one of the primary motivations of the study. For a while, it was thought that these earliest animals required relatively more oxygen than their unicellular ancestors. But then, of course, you have to know, okay, well, then how much oxygen do these early animals really need relative to the unicellular ancestors? And we don't know. And that's actually one of the primary motivations of this experiment that we did.
Obviously, one challenge you must face is that unlike someone who's, say, a paleontologist who is studying dinosaur remains and they have something physical to look at, you're trying to build a picture of things that were around hundreds of millions of years ago. There's nothing left of them except perhaps a genetic legacy. So is that the direction you've taken? You're looking at the way in which their genes are or aren't present in what's around today to try and work out what they must have been like, at least genetically speaking.
That's exactly right. So we're particularly interested in how animals respond to changes in environmental oxygen availability. Previous studies have identified this pathway called the hypoxia-inducible factor pathway. And hypoxia-inducible factors are just proteins that respond to oxygen. And this pathway essentially initiates genetic changes that the animal uses to deal with low oxygen. For example, production of red blood cells or new blood vessels, or even ways of producing energy without oxygen in the first place. Prior to our study, this particular mechanism of dealing with low oxygen had been identified in all animals surveyed. But these previous studies left out two vital animal groups, comb jellies and marine sponges. And these animals are thought to be some of the first animal lineages to split from our common animal ancestors. So they're at the very base of the animal tree. And no one had ever looked at whether or not sponges and ctenophores have this oxygen response pathway. So if sponges and ctenophores have this pathway, that would suggest that the last common ancestor of all these animals also had this pathway. And it's from the common ancestor that all modern animals have inherited this mechanism. But if sponges and ctenophores don't have it, then it suggests that this was not actually present in the earliest animals that evolved later on.
So in other words, if those animals do have this oxygen-sensitive pathway, these very primitive animals, that would argue that it was oxygen that was holding everything back. And if they don't have that, then it argues that perhaps something else, not just oxygen, was slowing down the rate at which multicellular animals appeared on Earth in the first place.
If sponges and ctenophores had this pathway, it would at least suggest that maybe this pathway was a necessary precondition for the evolution of modern animal life as we know it, and that until they figured this out, this oxygen response pathway, that animals as we know it couldn't have evolved. But then alternatively, right, if these other animal lineages don't have it, and it wasn't present in the last common ancestor of all animal life, it was not a necessary precondition for animal life as we know it.
So the $64,000 question, or perhaps even more valuable than that, do they have that function or not?
Sponges and ctenophores do not have this function. So we only have genetic data from ctenophores that suggests that they lack it. But with sponges, not only do we have the genetic evidence suggesting that they lack this pathway, we have experimental evidence essentially confirming it.
How did you prove it, though? How do you know that they may not have the same genes, for example, that you and I have, switching on when we see a low oxygen environment? How do you know that they haven't got equivalent other genes which produce the same end result but actually are a little bit different?
So we took marine sponges and put them in a setup in the laboratory that exposed them continuously to oxygen depleted water. We noticed that they weren't changing their genetic expression. If they had this pathway, you would have expect a shift in the sorts of genes that they were turning on and off. But we didn't see that response at all. There's essentially no difference between the sponges that we looked at under low oxygen compared to the high oxygen controls.
So it's not just oxygen which explains why we're here. Some other factors must also play a role. Daniel Mills there, looking back 1 billion years into life's origins. About 1% of children are diagnosed with the neurodevelopmental disorder autism. This means, among other things, they struggle in social situations. But is this something that develops or worsens because they have autism? Holger Spurdin and Marie Scher have been looking at children diagnosed as autistic or on the autism spectrum to find out how information is processed in their developing brains. Their results suggest that those able to compensate tend to perform better. So could a paucity of social information flow early in life, they're wondering, be one of the reasons why the brains of individuals with autism subsequently develop more abnormally? And might this also point to a better way to help those at risk to maximise their potential? Holger first.
When we talk about autism, infants that are later diagnosed with autism at, let's say, 3 or 4 years, they are less attuned to social cues. But we didn't have any kind of evidence for that in the brain, so we didn't know if there were alterations in the brain or differences in the brain that were already evident at such a young age. So the idea here was to explore that question. So basically, how does the brain physiology of very young children that have been diagnosed with autism, how different is their brain or not from that of typically developing children?
How did you actually do the study? What was the method here?
So what we did in this study, we studied really young children, so toddlers and preschoolers aged between 21 months and 48 months. We had in total 36 participants, and we compared 2 groups. One group that contained 18 children with ASD or with autism, and another group that didn't have autism. The techniques we used were eye tracking that allowed us to precisely see where those children were looking at. And to see what was going on in their brains and to compare between the groups, we used what we call electroencephalography, or EEG for short.
So what did you show the children, and how is that reflected in the EEG data?
What we showed to them was 2 video sequences of dynamic social images that had a duration of about 4 minutes. They included other young children that they had never seen before practiced, for example, yoga movement alone. They were also imitating animal-like behaviors, behaving like a monkey, or they were jumping like a frog. They waved their arms, made faces. So really lively stimuli of other children moving around. What we first found, toddlers and preschoolers with ASD, they were exploring quite differently those videos. And those differences were also reflected inside the brain. And the first notable finding was that how information flowed in their brains was very different from one area to another in the brains of the children with ASD compared to the typically developing pairs. That was the first finding. And the second one is then that we correlated, for example, the way the children with autism were looking at those dynamic scenes with those different metrics we had from the EEG. And what we found was that we had a much stronger brain response, if you want, in the brain of those who were less severely affected. So the hypothesis we make here is that some children, I mean, we don't still understand why, but they manage to compensate that way, you know, in the long term. So let's say that if ASD kicks in at 3 or 4 months after birth, they had 2 years to kind of, you know, reorganize or rewire the brain until they are 4. And this is as opposed to those without ASD, of course.
Do you think if you had done the study longitudinally, you'd taken the same children, same stimuli, and looked over a period of time, do you think what you'd see is this pattern becoming more entrenched as they got better at responding to social cues? This is the autistic children I'm referring to.
Exactly, Chris. So this is something that we are currently pursuing in the lab. So we are following up those children. So they are coming each 12 months. So we do 2 extra recordings in the same conditions, right? And what we want to look at is how those that are, following early intensive intervention or community intervention, as opposed to those who don't have any interventions, for example, how those different metrics we found in the brain, how they evolve over time. So for example, let's say those who were more affected in this particular study here and who have, for example, a less strong brain response as opposed to those who are less severely affected, will they start also to show us this same pattern after treatment, let's say 12 or 24 months after? And we think that this might be the case.
Holger Sperdin. Mary Scher, who led the project, thinks their findings might point to better ways to manage young children who are at risk from autism.
Those children from the age of a few months old, they attend much less to the social stimuli. So their brain is getting less experience about all those social stimuli. And it means that already very early at the age of 2, which is exactly when we diagnose them, they probably already shaped their brain in a way that is already very different, but it's only at that time that we can diagnose autism.
Do you think that you might be able to use the learning from this study to come up with strategies that mean you can intervene more meaningfully than we currently do in order to help people to become less severe on that spectrum?
One of the particularities that we had in our study is that we had eye tracking information at the same time as the brain activity. That means that we knew exactly what the children were looking at. And what we saw is that the one who looked at the information in a more typical way were also the one who had a more typical brain development. So one of the hypotheses for the future research is for us to look whether treatment in which we increase the social skills of the children, such as early start Denver model or other type of treatments that are really dedicated to develop their social skills, will then affect the brain over this very critical period of the age between 2 and, let's say, 5-6 years old.
Mary Scher and before her Holger Sperdin. They're both at the University of Geneva. That's it for this month. You've been listening to the eLife podcast, previous editions of which are also on eLife's website. That's elifesciences.org, or you can subscribe on iTunes. To help you to follow up on the discussions you've heard, there are also some text transcripts of each of these programmes with the supporting references to the published papers on the Naked Scientist website at nakedscientist.com/eLife. I shall be back with another look inside eLife next month. Until then, though, thanks for listening, and from me, Chris Smith. Goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.